All right, today we're doing something a little different. We're going to deconstruct the science of how we see the invisible world. And our source material, it's not a book, it's not some article. It's actually a university exam paper on microscopy. So let's crack it open and see what it can teach us. So let's kick things off with a bit of a riddle. What on earth could a living, blob-like amoeba possibly have in common with a human nerve cell, which is one of the longest cells in your entire body? Well, the answer is all about what we can't see. With just our eyes, both of them are pretty much invisible. They exist in these tiny, hidden worlds that we can only ever hope to explore if we have the right tools to get us there. So here's our game plan. We're going to follow the roadmap laid out in this exam. We'll start with why there's more than meets the eye, then get into the really crucial difference between making something bigger versus making it clearer. We'll look at the amazing toolkit scientists have, uncover some of the built-in flaws in our best instruments, and then we'll tie it all together to see why any of this matters. All right, first up, more than meets the eye. This is really the first giant hurdle that biology as a science had to overcome. The simple fact that so much of life happens on a scale that's just way too small for us to see on our own. So the most basic idea, and it pops up all over this exam, is magnification. It's really just a simple ratio, how big something looks compared to how big it actually is. It's the core concept, right? Making a tiny thing look huge. But as anyone who's ever zoomed in way too far on a photo can tell you, bigger is not always better. And that brings us to the real secret sauce of microscopy, the constant battle between making things bigger and making them clearer. Think about it like this. You can blow up a fuzzy, low-quality picture to the size of a billboard, but all you'll have is a giant, fuzzy, low-quality picture. Making it bigger? That's magnification. But the real magic, the thing that actually shows you the details you're looking for, that's resolution. That's what makes things clearer. So what exactly is resolution? Well, the exam gives us the official definition. Imagine two tiny specks of dust sitting incredibly close to each other. Resolution is the ability to actually see them as two separate specks, instead of just one blurry blob. It is quite literally the definition of detail. So, how do we get more of it? How do we see those finer details? Well, the exam points us right to the light itself. It turns out the color of light you use, its wavelength, is absolutely crucial. Using light with a shorter wavelength, like blue light, is kind of like using a finer, sharper pencil. It lets the microscope draw a much more detailed picture. And this is where the scale of what we're talking about just gets mind-boggling. The exam gives the limit for a good light microscope as 240 nanometers. To put that in perspective for you, a single strand of your hair is about 80,000 nanometers wide. We are talking about seeing details on a scale that is just almost impossible to imagine. But what if even your best light microscope just isn't cutting it? Well, that's when you have to break out the specialized toolkit. Because seeing different invisible things requires totally different strategies. It's definitely not a one-size-fits-all situation. The exam paper lays out these tools like solutions to very specific problems. Got something that's almost totally transparent? You use dark field microscopy to make it pop against a black background. Need to see the inner workings of a cell while it's still alive? Phase contrast is your go-to. And if you want to pull off a real magic trick, like making one specific protein literally glow in the dark, you turn to fluorescence microscopy. And check this out. This diagram is pulled straight from the exam itself. This is a simple blueprint of that very tool, the fluorescence microscope. It looks a little complicated, I know, but you can think of it as a really clever system of filters and mirrors, kind of like bouncers at a nightclub for light. So how does it work? Okay, imagine you've written a secret message on paper with invisible ink. In normal light, you see nothing. But if you shine a black light on it, the message suddenly glows bright green. This microscope does exactly that, but on a molecular level. It shines one very specific color of light onto the cell. Everything ignores it except for the one tiny molecule we've tagged with a special fluorescent dye. That molecule absorbs the light, and then it glows back with a totally different color. The whole system is designed to block the original light and only let that glowing secret message light get to our eye. So all we see is the one tiny part we wanted to light up. But even with these amazing, almost magical tools, the hunt for a perfect image is never quite over. And that brings us to the problem of the imperfect lens. Turns out, the very physics of how lenses bend light creates built-in flaws, little glitches that can warp the very reality we're trying so hard to see. The exam paper calls out a couple of these glitches, which are called optical aberrations. With spherical aberration, light that hits the edges of the lens doesn't focus in exactly the same spot as light hitting the center, and that causes blur. Then you've got chromatic aberration. A simple lens bends different colors of light just a little bit differently, kind of like a prism making a rainbow. The result is that all the colors don't focus together, which gives you those fuzzy, color-fringed edges. And this quote, which is pulled right from the exam, just nails the problem perfectly. When light from your sample isn't brought to a simultaneous focus, you lose sharpness, you lose clarity, you lose all that precious detail you were working so hard to see. Modern microscopes, by the way, use incredibly complex combinations of lenses just to try and fix these exact problems. Okay, so we've looked at the fight for a bigger, clearer picture, and the incredible tools we've built to get there. But so what? What is the point of all of this? And that brings us to the big picture: why seeing matters. The answer, believe it or not, can be found in this one simple, powerful statement from the exam. Cell theory, the fundamental rule that all living things are made of cells, does not apply to viruses. Now, how could we possibly know that for sure? Simple. We know it because we can see them. Our ability to build tools powerful enough to actually visualize a virus is what allows us to write the fundamental rules of biology, like cell theory. And maybe even more importantly, it allows us to discover the weird, fascinating exceptions that are constantly pushing the boundaries of what we even consider life. In the end, this is really the story of science itself. Every huge leap forward in our understanding has come right after a huge leap forward in our ability to see. From the simplest little lenses to glowing molecules, we've basically built incredible extensions of our own senses. And it just leaves you wondering, what other invisible worlds, what new rules of life are still out there, just waiting for us right beyond the edge of what we can currently see?